Assalamu alaikum everyone. This is me, Dr. Faiza here, with the third session of Special Senses. Topic here today we have Central Neurophysiology of Vision. This is the last session on the physiology of optics. Till now we are done with the basic principles of optics, intraocular fluids, errors of refraction, and receptor and neural functions of the retina. Today we are going to move towards the central neurophysiology of the vision. For that, I have used references from the standard textbooks, majorly from the Guyton and Hall, and rest of the references are given in front of you. To begin with our discussion today, first let's have a view. What is the field of vision? If you are glancing at something, whatever you can see at an instance, that is your field of vision. If you close one of your eyes, this is the monocular field of vision. If you open both of your eyes, this is your binocular field of vision, which is obviously more than as compared to the monocular field of vision. For both the right eye and left eye field of vision, the field of vision can be further divided into a nasal half and a temporal half. Nasal half and a temporal half. So we are going to now talk about the neurophysiology, how from the retina via the optic nerve the message is carried to the visual cortex. We all know optic nerve arises from the ganglion cell axons at the level of retina and exits the retina at the level of the optic disc. There is one optic nerve for the right eye and another optic nerve for the left eye. In front of you, the diagram is showing this is the right eye and this is the left eye. For both, the field of visions is divided into temporal half and the nasal half. We had decided in the first session that the retina image is reversed, upside down, and also reversed right and left side. So the nasal half of vision is actually represented on the temporal part of retina, and temporal field of vision is actually represented on the nasal half of retina. The superior field of vision is represented on the inferior part of retina and the vice versa. So this is again the diagrammatic representation of this reversal. The nasal half of vision is carried upon by the temporal fibers of retina, whereas the temporal field of vision, the message is carried by the nasal fibers of the optic nerve. Once the nerve leaves from the retina, it has representation of both field of visions, nasal and temporal field of vision. It has both the fibers from the nasal half of retina and the temporal half of retina. Initially, the right and left optic nerve are responsible for representation of the vision from solely right eye and the left eye. After a while, their nasal fibers from both the optic nerves cross each other. They cross over. You can see at this level of B, optic chiasm level, the nasal fibers have crossed over, whereas the temporal fibers of retina are still running on the same side. After this crossing over, optic nerve is now called as optic tract. Optic tract has nasal fibers from the contralateral retina, nasal half of left retina, this right optic tract, and temporal half of the same side of retina, that is right retina. This optic tract goes on and synapses with the lateral geniculate body. This is the dorsal lateral geniculate nucleus of the thalamus. So this synapsing occurs at the level of right side and left side of lateral geniculate body from the right and left optic tract. Again, after relaying, the radiations come out and move to the visual cortex. Again, these optic radiations, now these are called as optic radiations. Optic radiations again have the representation from the temporal retina of same side and nasal retina of the opposite side. But there is one more demarcation which was not present in the optic tract. At the level of optic radiations, the nerve fibers from the upper half of retina are separated from the nerve fibers from the lower half of retina. What happens? The fibers from the lower part of retina, that is representing the upper field of vision, they move through the temporal lobe and they end in the visual cortex at the lingual gyrus. As you can see here, the lingual gyrus termination moving through the temporal lobe, representing the lower retina or the upper half of field of vision. This is called as Mayer's loop. Whereas the retinal fibers from its upper half, representing the lower field of vision, move through the parietal lobe and they end at the cuneus lobe of the visual cortex. So one thing more happened here is that the upper and lower field of vision is also demarcated at the level of the optic radiations. Once they reach the optic cortex, now the right optic cortex will be representing the temporal half of right eye and nasal half of the left eye. If we see one by one at all these levels, what the field of vision is represented, the optic nerve of the left side. Let's have a look on the left side of these visual pathways now. The left optic nerve will represent the field of vision of completely the left eye, nothing of the right eye. At the level of optic chiasm, the central fibers, they will be representing nasal half of both retina. Nasal half of both retina means the temporal field of vision of both eyes. So this is temporal field of vision of both the right and the left eye. At the level of optic tract, left optic tract has temporal fibers of left retina, means nasal field of vision of left retina. Secondly, they have nasal fibers of right retina, means temporal field of vision of right retina. So they will represent this. Left optic tract is representing left field of vision of both eyes. Optic radiations are doing the same, but if we look at both the superior and inferior quadrants separately, the fibers in the Mayer lobe, they will be representing the upper field of vision, and those going to the through the parietal lobe, they will be representing the lower field of vision and the contralateral half. So after optic chiasm, it's contralateral. Right optic tract, left field of vision, left optic tract, right field of vision of both eyes. If we go to the optic radiations, it is further demarcated into superior and inferior, and now this is not the half, this is the quadrant. Once we reach at the level of visual cortex, they will be having the representation from both the upper and lower radiations. So they will now be representing the contralateral half. Left visual cortex will be representing the right field of vision of both the eyes. You can see some donut-like appearance over here, central portion. This is the macular representation, which is the bilateral. Both the visual cortex will have representation of macula latia from both the eyes. So that's all about the visual pathway. If you are asked a question about the visual pathway, think wisely. If you are asked about the retinal fibers or about the field of the vision, mostly you are asked in context of the field of the vision. Which field of vision is represented by right optic tract? Which field of vision is represented by the right optic radiation, by the optic chiasm, by the left visual cortex, by the Mayer lobe? If you know the anatomy, you can answer all these questions very easily. Also, you can answer what will happen if there is a lesion at any level of this optic tract. What you need to focus on is to focus on the anatomy and remember the rules of reversal. The right eye nasal half of retina will represent the temporal field of vision. Temporal retina will represent the nasal field of the vision. Now that we have seen the visual pathway, let's talk about the defects in the visual field. Smaller defects. Blind spot. We had seen that in our second session on special senses. One of this is the physiological blind spot, which is found at the level of the optic disc. We all have that physiological blind spot, but the physiological blind spot of our one eye is covered by the other eye, so we do not feel it, but you can find it out by certain tests where your actual blind spot is. Pathological blind spot is the blind spot at a point in the retina other than the optic disc. Anywhere other than the optic disc, this will be pathological, and the other name of this pathological blind spot is scotoma. Then blindness, anopia, right anopia, left anopia will be complete blindness of the right eye or the left eye. If we say hemianopia, that means this is the defect in one half of the vision, either temporal or the nasal half. Hemianopia. This is further of two types, homonymous and heteronymous. If the right field of vision or left field of vision of both eyes is lost, this is homonymous, right and left homonymous hemianopia. Something like this. This is black one is showing the blindness, the loss of vision, and gray one is the normal vision. So this is showing right homonymous hemianopia. If it would have been left homonymous hemianopia, left field of vision of both eyes would have been lost. On the other hand, heteronymous hemianopia is that for both eyes, different sides of field of vision are lost. This can be called as heteronymous hemianopia or bitemporal hemianopia. Temporal fields of vision of both eyes are lost, or this could have been by nasal hemianopia, that nasal field of vision of both eyes is lost. Both are hemianopias. If this is different for both eyes, right or left, this is heteronymous. If right or left field of vision of both eyes is lost, this is homonymous hemianopia. If we go further at the level of optic radiations, if the lesion occurs, the field of vision lost might not be the complete half, but just one quadrant. It can be superior or inferior quadrantanopia on the right side or on the left side. If the quadrantanopia is on the same side of both eyes, right half of vision, superior field of vision. This is right, superior, and homonymous quadrantal. Right, inferior, homonymous quadrantanopia. If it is occurring on the different sides of both eyes, one on the left side and one on the right side, now this is superior heteronymous quadrantanopia. This is rare. So let's stick to the homonymous types. Now that you know the physiology of the visual tract, how the fibers go from the retina to the visual cortex. If you are asked about the lesion at any level, you must be able to guess what the effect will be. So let's start guessing. If the lesion occurs at this level, this is left eye and this is right eye. Left optic nerve is completely damaged. What will happen? There will be complete blindness of the left eye. As you can see over here, anopia of the left eye. If lesion occurs at the level 2 that you can see over here, here only the temporal fibers are lost, temporal retinal fibers of the left eye. That means nasal field of vision of the left eye So left nasal hemianopia. Right eye is fine. Temporal field of vision of left eye is also fine because only temporal fibers of retina were damaged. Number 3, damage at the level of optic chiasm and only the central nasal fibers are destroyed. If only the nasal fibers are destroyed, that means temporal field of vision of both eyes is lost. That you can see over here. Temporal field of vision on both eyes. This is called as bitemporal heteronymous hemianopia. Moving on, level 4. Here the left optic radiation is disturbed. It has temporal fibers of left eye, means nasal field of vision of left eye, and nasal fibers of right eye means again the temporal field of vision of the right eye. So the picture you will have is this. On both sides, right half of vision is lost. Nasal here, temporal here, but it is right on both sides. So this will be right homonymous hemianopia. You can appreciate if you know the visual tract anatomy, you can guess the effect of lesion at any level. Number 5 is the lesion to the Meyers's loop that was having the representation of lower retina, means the loss will be superior field of vision. Number 2, it has fibers from the left temporal and right nasal retina. It will be leading to the homonymous type of loss on the opposite side. So right homonymous superior quadrantanopia. If the fibers via the parietal lobe are disturbed, again it will be opposite side homonymous anopia, but on the lower half of field of vision. Right homonymous inferior quadrantanopia. Once we reach at the level of the visual cortex, if damage is in the left visual cortex, again contralateral hemianopia, homonymous hemianopia, right homonymous hemianopia. But one thing is added to this, which is macular sparing. Why macular sparing? As we discussed in the previous slide, macula is represented bilaterally for both the eyes. So this will keep the macular vision safe. The fibers reaching the left visual cortex are from the temporal half of left retina and nasal half of the right retina. Temporal half of left retina and nasal half of right retina. This is retina, left and right. If we translate it into the field of vision, it will be nasal field of vision here and temporal field of vision here. If we translate it into these circles, loss will be nasal field of vision of left eye and temporal field of vision of right eye. If we translate it into right and left, this will be right on both sides, because right on both sides we will call it homonymous because it is involving half of the square. This is hemianopia. So if you have a question, first of all think about what fibers it is carrying, temporal or nasal from both the eyes. Then translate it into the field of visions just by reversal. Then fill in your circles and see if it is on the right side, on the left side, bitemporal, binasal, and then name it. If you think sequentially, you will never miss the diagnosis. One thing more, if you want to make it even easier for you in the exam, you solve this question and you are still unsure if you have done it right or not, you can see after the optic chiasm, when the crossover has taken place, the loss is always going to be of homonymous type. Left-sided lesion, right homonymous hemianopia. right homonymous quadrantanopia, again right homonymous hemianopia. Optic radiations, optic tract, hemianopia, optic radiations, quadrantanopia, cortex, again hemianopia with the macular sparing. Heteronymous hemianopia is only going to be found at this level, at the level of optic chiasm. Before that, above it, till the optic nerve, it is going to be anopia. At the level of optic chiasm, it is always going to be heteronymous. And after that, it is going to be homonymous hemianopia of opposite side. Anopia will be of same side. Hemianopias will be of the opposite side. Heteronymous hemianopia is mostly asked of the central fibers, which is bitemporal heteronymous hemianopia. It's very rare to have binasal homonymous hemianopia because this is a rare injury that will damage the temporal fibers while leaving the nasal fibers. Usually either it is a complete loss or only the central fibers because of the pituitary tumors that you will relate it in your endocrinology. But this is very rare to have an injury of only the temporal fiber. So it's very unlikely that you will have a scenario of heteronymous binasal hemianopia, but this is very common. Bitemporal heteronymous hemianopia, very, very common, and rest are all homonymous hemianopias on the opposite side. Not only the visual fibers originating from the retina go to the visual cortex, but they have some other accessory functions too. Some of the fibers from the ganglion cells having melanopsin, instead of scotopsin and photopsin, they leave the visual tract and enter the suprachiasmatic nucleus of hypothalamus to control the circadian rhythm. Some of the fibers go to the pretectal nuclei at the level of optic tract. The fibers leave and enter the pretectal nuclei in the midbrain. They are involved in one, reflex movement of the eyes, and secondly, in the pupillary light reflex. Some of the fibers from the optic visual pathway, they go to the superior colliculus, which is involved in controlling the directional movement of the eyes. And lastly, instead of dorsal lateral geniculate nucleus of thalamus, some might go to the ventral lateral geniculate nucleus of the thalamus, which may be involved in the body's behavioral functions. Few of these details we'll be seeing in the coming slides. It is also said that the fibers going to the superior colliculi, pretectal nucleus, and ventrolateral geniculate nucleus, this is the old visual system, which is not in under voluntary action, and the rest we see that is going to the visual cortex is the new developed system. As we talked about the dorsal lateral geniculate nucleus of the thalamus, which is the lateral geniculate body, here the optic tract terminates and optic radiations arise. So this is first of it, its relay function. Lateral geniculate body is receiving signals from both the eyes, temporal half of one retina and nasal half of the other retina. It keeps it coded likewise. It does not get it mixed. Rather, out of the 6 layers, 3 layers will be receiving from the same side of the eye and other 3 will be receiving message from the contralateral retina. Layer 2, 3, 5, 1, 4 and 6, and this representation goes on till the visual cortex. Starting from the optic tract, when the message was mixed with the right and left eye, still the message is not intermingled. It is kept separated in the layers of thalamus and also in the layers of the visual cortex. Another function performed by the thalamus over here is its gating function. Not every message is passed to the visual cortex, otherwise there will be bombardment of messages on our visual cortex. There are inhibitory corticofugal fibers coming from the visual cortex back to the thalamus and from the other areas of the brain to filter the messages which are important to be sent are sent and rest are inhibited. As we saw the division of ganglion cells into parvocellular and magnocellular ganglion cells, this demarcation is maintained at the level of dorsal lateral geniculate nucleus of thalamus too. The parvocellular pathway and magnocellular pathway starting from the ganglion cell to the thalamus to the visual cortex, this is maintained. Parvocellular pathway will be concerned with more of the color vision. It will be having slow conduction velocity. It will be having low sensitivity but more accuracy. They will be receiving input from the P-type retinal ganglion cells, and these are found layer 3 to 6 of the lateral geniculate body. Now this is different from this. Whereas magnocellular pathway is going to relay the message at layer 1 and 2 of lateral geniculate body. It has more sensitivity, faster conduction, involved with black and white vision, and they are going to receive input from the M-type retinal ganglion cells. So same characteristic carried forward from the ganglion cells to the lateral geniculate body, and same will be carried to the visual cortex, and the pathway will be demarcated as parvocellular and magnocellular pathway. If there is rapid movement of some object across your eye, you will detect it instantaneously because of the magnocellular pathway, but you will not be able to detect the particular characteristics of that object because parvocellular pathway has low sensitivity. It cannot respond to rapid change of the stimuli or low intensity stimuli. Once the message reaches the visual cortex, what the visual cortex is, where is it found? So primary visual cortex, also known as visual area 1 or stria cortex because of the multiple stria to increase surface area over here. Goodman area classification you can see over here is 17. It is found in the occipital lobe, calcarine fissure area, and it moves forward on the medial aspect of each occipital cortex. In front of it is the visual association area. In the primary visual cortex, you can see further demarcation of the messages. The posterior most is the representation of macula lutea, and this is the having the largest representation of the fovea centralis. Again, like that of the sensory and motor homunculus, which is having the highest density of innervation and highest acuity, highest precision, is having more representation in the cortex. Here the macula and fovea centralis are having the highest representation, moving onwards from the other 5 areas of the retina. Away from the macula, the more away it is, more towards the periphery, more anteriorly, it will end in the primary visual cortex. In front of the primary visual cortex is the secondary visual areas. Again, the difference is maintained that upper portion of retina is represented superiorly. It will receive fibers from the upper portion of retina and will represent the lower half of field of vision and vice versa. Secondary visual areas are many, not just 1, 2, 3, 4, up to a dozen secondary visual areas which are involved with different functions. We are not supposed to memorize the functions of all these secondary visual areas one by one, but I'm going to show you this, that there are many secondary visual areas associated with different interpretative functions of the visual message. For example, motion, detection of shape, color, direction of movement, speed, recognition of shape, representation of upper lower quadrants of visual field. Do not memorize it, but make a mind map that how all these specialized areas are helping in the interpretation of the visual message at a higher cognitive level. But do remember the last one, the V8. For color vision, we will be seeing why is it important. We saw in the yesterday's session that achromatopsia, color blindness, can be caused by the defect in the visual cortex, and this is the portion of the visual cortex, if defective, can lead to achromatopsia. Looking into the further details of primary visual cortex, like the cortex in other part of the brain, the visual cortex is also having 6 layers. Input is majorly coming from magnocellular and parvocellular pathway to the layer 4. The magnocellular pathway coming from the M type of ganglion cells, which has higher sensitivity, is going to majorly terminate in the layer C alpha, whereas the parvocellular pathway is majorly going to terminate in layer A and C beta. From here, the message is sent into the layers upwards and downwards. Later on, the message from the visual cortex leaves via layer 1, 2, and 3 to the nearby areas, and layer 5 and 6 to the areas away from the visual cortex. Neurons in the layer 4 have no preferred orientation. They will respond to the visual stimuli of any size, shape, orientation. These are less complex. Moving up or down, specifically layer 2 and 3, the complexity of the receptor functions increases, like that of ganglion cells and bipolar cells. Visual cortical cells also exhibit the phenomena of on center, off surround, off center, on surround, defined by the lateral inhibition, which increases the ability to make a sharp image on the retina. The magnocellular pathway and the parvocellular pathway. We have seen it from the ganglion cells to the geniculate body and to the visual cortex. Now this slide is showing from the primary visual cortex. After the message is received, the message after interpretation is sent to the surrounding areas of the brain. Those involved with magnocellular pathway interprets majorly the position, form, motion of the body, and also it is involved in stereopsis, the depth perception we saw in the first session. From here, the message is sent to the dorsal and parietal areas of the brain. Whereas from the parvocellular areas pathway, which is involved with the visual details, color vision, texture, shape, the message is sent to the ventral and temporal areas of the brain, because every sensation is ultimately going to be collaborated with other sensations to make a meaningful understanding for the person. This is a beautiful diagram showing what we are looking at. This is the retinal images which is formed. If we, for example, look at this plus sign, this retinal image will be formed. But because of the phenomenon of on-off surround center phenomena, lateral inhibition, the ultimate image that forms at the level of cortex is like this. So actually, the eye is not looking at something very solid. The brain is looking at the sharp contrast borders. The more the gradient of this contrast is, more sharp and clear the image will be formed. So maximal cortical stimulation will be happening at this when there is a change in the contrast. Rest of it will be ignored by the cortex. One of the functions which will be performed at the level of geniculate body, where cortical fugal fibers will be coming to ignore the function stimuli which are less important. Moving on towards the little complicated topics, let's try to make it easy for you. Color blobs. Its name indicates. Its name shows that this is somehow involved in the interpretation of the color vision. In front of you is the visual cortex, which is showing alternation of right eye and left eye. This is, we know, messages coming from both the eyes alternatively at the geniculate body, and that goes on to the visual cortex. Next thing you can see, these are orientation ocular dominance columns, which we will be looking at in a while. Third thing you can see are the color blobs. Color blobs are basically the group of neurons which are specifically sensitive to the color vision. They arranged in cylindrical shapes inside the visual cortex, and these are present in layer 2 and 3. Although the parvocellular pathway neurons were terminated in layer 4, The message is also sent to color blobs in the layer 2 and 3. Their particular histological characteristic is having them a high concentration of cytochrome oxidase enzyme in their mitochondria. So color blobs are the group of neurons in layer 2 and 3 specifically involved in the interpretation of the color vision. If we look at the layers of the visual cortex, there are different type of cells which act as feature detectors. Simple cells are simple. They respond to any shape, any type of visual stimulus. They have no specific preference. At any shape, at any angle, of any size, the simple cells will respond. Then comes the complex cells. Mostly these complex cells are present outside the layer 4. For example, in layer 2, 3. What their complex cells do, they are stimulated only when the visual stimulus comes at a specific angle. For example, a visual stimulus at this, a visual stimulus at this. So for every angle in the 3D world, there are complex cells which will respond to that particular angle and they will be activated. They have preferred orientation, but they are not location specific. If that visual stimulus at 30 degrees angle is found in the temporal field of vision, nasal field of vision, superior or inferior quadrant of vision, that does not matter much for them. Location does not matter. For simple cells, location was what mattered. For complex cells, the orientation is what matters. Location is the less important thing for their stimulation. So if there is a line at this angle, a pen at 30 degrees, my complex cells will be activated. Even if the pen moves from this to this half of vision, those complex cells will still be activated. In comparison, simple cells are activated if this pen is in my field of vision at 90 degrees, 30 degrees, 60 degrees, 50 degrees, does not matter to them. And then there comes even more complex cells, which we call hypercomplex cells. They are not dependent upon the location of the object or the stimulus. They are not dependent on the even orientation of the subject. They are dependent upon the shape. They will be stimulated only by the specific lens shape border. For example, a star shape of this size, hypercomplex cells will be activated only when that comes into my visual field, in whatever part of the field of vision at whatever angle. So this is some additional information that you might skip even. Orientation columns. What are these orientation columns? These are mostly outside the layer 4. They are flat columns perpendicular to the surface of visual cortex. If this is visual cortex, these columns will be present perpendicular to it and they will respond to visual line stimuli of varying angles. Like the complex cells which responded to orientation, these visual columns will respond to certain orientation. As you can see in this diagram, different angles orientations are shown. Irrespective of the location of the stimulus, the orientation of the stimulus will stimulate these columns. So for each ganglion cell in the receptive visual field, there is a collection of the orientation columns. For example, this is the visual field of right eye and this is visual field of the left eye. If there is a visual stimulus at 50 degrees here, certain sets of orientation columns will be activated. If the stimulus is at the level of 30 degrees, different sets of orientation columns will be activated. So this will help the brain in deciphering that where the image is actually in the visual field, depending upon which orientation columns are activated. Ocular dominance columns, their name indicate the Visual cortex is receiving message from both the eyes. So there will be certain cortical neurons which will respond more to right eye and certain neurons which will be responding preferentially to the left eye. That forms the ocular dominance columns, showing their dominance with the right and left eye. And if we look at the ocular dominance columns from the top, it will be forming something like a mosaic, alternating right and left dominance. We are talking about field of vision continuously. We said that it's a visual area seen by the eyes at a given instant, nasal half, temporal half. Clinically, you can evaluate it with the help of perimetry. You will also be doing it in your physiology labs. What it shows is you can see this is field of vision, and this is more on left side of the eye, temporal side of the eye. This is temporal field of vision. If I am not given any further information, I can guess this is temporal. And this is nasal field of vision, because on the nasal side, this is obstructed by the nasal bridge. This is maximum on the temporal side. Superiorly, it's obstructed by the rim of my frontal bone, and inferiorly by the cheek prominences. So the maximum visual field is going to be on the temporal and also on the inferior side. You can compare superior, inferior, temporal and the nasal sides of the visual field. This wide area is showing the visual field of the left eye. One thing more you can see over here, this pink spot. This is the blind spot. If you are doing perimetry in your labs, you can all mark your blind spot at these perimetry charts. You can find it out, which on average is 15 degrees lateral to the central point of vision. This is central point of vision and this is 15 degrees lateral to it. And this is on the horizontal meridian, usually with little bit of variation. This is optic disc, which will be the physiological blind spot. Once we discussed the optic disc in our second session, we said that this is just this is posterior point of retina, which is fovea centralis, and just medial to it and little above it is found the blind spot. Now the blind spot or optic disc is on the medial half of retina and lateral field of vision. You see the reversal rule. So if you are asked a question about it, do see if you are asked about the retina or field of vision. On retina, this is just medial to the fovea centralis, and on the field of vision, this is just lateral to the central point of vision. This is a scotoma. As we discussed, any blind spot other than the optic disc will be pathological, and that can be known as scotoma. This can be caused by the damage to the optic nerve fibers by many reasons, one of which we discussed in our first session, glaucoma. This is showing macular degeneration. We discussed very clearly that fovea centralis macula lutea has the highest visual acuity, central field of vision related for some reason. For example, aging can lead to age-related macular degeneration, or other comorbidities can do that. In that case, central field of vision will be lost, whereas the peripheral field of vision will be still working. This is the opposite of it. Here, the central field of vision is fine and peripheral field of vision is defective. One of the reasons can be retinitis pigmentosa, a characteristic cause of this tunnel vision, which is caused because of excessive melanin pigmentation. So that was about the visual pathway and its defects. Let's have a quick review of the movements of eye. You will be doing the movements of intraocular muscles, the medial longitudinal fasciculars, mostly in context with your cranial nerves and anatomy. I'm hereby going to focus more on the types of movements of the eyes important from the physiological point of view. So let's move on to that. Ocular movement. First of all, conjugate eye movements. If you have to see rightwards, one of your eye will move towards lateral side and other will move towards its medial side. If both move towards opposite side, you will not be able to focus on something. So the conjugate eye movement is necessary. and that is made possible with the help of the medial longitudinal fasciculus. Important eye movements that we are going to quickly review right now include the fixation, pursuit, vestibular, and convergence eye movements. Starting from the fixation eye movements. You want to fixate on something. You want to look at this pen, and you want to, number two, look on this glass. So what you will have to do is fixate your gaze on this pen for a while. Interpret that this is a pen, then unlock your gaze and move it to this glass. And lock your gaze again at the glass. If you want to look at the pen again, you will unlock your gaze from the glass and move it to the pen and fix it again. So fixation movements need both these things, locking and unlock. So different centers in the brain are responsible for that. Unlocking is voluntary. This is performed by frontal eye field. Whereas the locking, once you move to the object, the locking is involuntary, and this is performed by secondary visual association area and also by the superior colliculus. And a special example of successive fixation movements, very important eye movement, saccadic movement, in which the gaze shifts from one point to the other in your field of vision very quickly. For example, you are reading a book, you are reading its slide, your eyes are quickly moving their gaze from one word to next line, next line, next line. So you are continuously locking, unlocking, locking, unlocking, locking, unlocking. Here your frontal eye field, your superior colliculus, your visual association areas are helping you lock and unlock your gazes, while your brain cuts off the time in the transition. Your brain does not appreciate that you are moving, you are just looking at the words. This is the saccadic movement. Here your field of vision is static, your eyes are jumping from one point to the other point. Saccadic movement. Superior colliculus and frontal cortex are very important. If there is a defect in it, saccadic movement cannot happen. If there is defect in the frontal eye field, what will happen? You fixate your gaze on the pen, and now you want to fixate on my finger, you will be unable to unlock your gaze from the pen. What you will have to do, you will have to close your eyes, open and fixate on my finger. You will not be able to unlock your eyes from the gaze. If there is lesion of superior colliculus or secondary association area, your eyes will be unable to lock. It will be impossible to maintain your gaze at a point. And also the saccadic movements will be lost. Pursuit movements, on the other hand, are the movement of the eyes when something is also moving in your field of vision. For example, a bee, a fly, a bird is flying in the air and your eyes are following it. For after a while, your eyes will come at a velocity equal to the velocity of that flying thing and you will be pursuing it. Its name indicates. It's an involuntary cortical mechanism and cerebellum helps this too. Vestibular ocular movements, for example, vestibular ocular reflex, again, superior colliculus is involved in it, will help you maintain your gaze with movement, will help you maintain your balance too. Convergence movements, you want to read something close to you, both of your eyes converge to that point. Conjugate eye movements makes this convergence possible. Also, this is a part of accommodation, which we will be looking in a while. Moving on, you look from both your eyes, images formed at the corresponding points on the retina. Again, the example, if I want to look at this pen, I will move my gaze over here. I may not move my head, simply move my gaze over here. Both my eyes move to it, and the image is formed at a point that I look at it as a single pen. Diplopia does not occur, and that is made possible by the conjugate movement of eyes with the help of the medial longitudinal fasciculus. And if the conjugate movement of eyes are defective, what will happen? Both the eyes will not be able to make the gaze on a single point. They will not be able to make the image on the same corresponding points on the retina. One of the conditions we very frequently see defective conjugate movement of eye results in strabismus, squint, crossed eye, name of the same thing. In Urdu, we call it bhengapan. What happens is there is lack of fusion of the eyes in one or more visual coordinates, maybe vertical, horizontal, or rotational. One eye has fixated on what you want to see, and the other eye is wandering in another quadrant. That leads to strabismus. What happens? It starts with abnormal set of fusion mechanisms of the visual system for both the eyes. Ultimately, the child cannot fixate simultaneously on both from both eyes on the same subject. Resultant is one of the eye will fixate satisfactorily and other will fail to do so. If it keeps on happening, ultimately the eye which fails to do so will become the lazy eye, and that will lead to abnormal conjugate movements, that will lead to suppression of the vision in the lazy eye, resulting in greatly decreased visual acuity, amblyopia, exynosmia. So strabismus just started with this, may end with this and this. If taken care of well in time, strabismus may be avoided and visual acuity may be kept safe. A very important aspect from your exam point of view, from the clinical point of view, just by abnormal set of fusion mechanisms of both eyes, one eye unable to fixate on the object, other can do it, the lazy eye will lose its visual acuity ultimately. Secondly, the conjugate eye movements will be defective, resulting in squint. That was all for the conjugate movement of eyes. Let's move on to the next topic, that is autonomic innervation of eyes. This is the last topic for today's session. Autonomic innervation of eye is both from the sympathetics and parasympathetics. We have seen these in one context or the other in the previous sessions. Let's have a look at it. Pupillary sphincter muscles supplied by the parasympathetic muscarinic receptors, their contraction leads to meiosis. That is pupillary constriction. And if this receptor is blocked, the pupils will dilate. Minimum diameter the pupils can achieve by pupillary constriction by contraction of pupillary sphincters, parasympathetic activation is 1.5 millimeter. On the other hand, the dilator muscles for the pupil, the radial dilator muscles are supplied by adrenergic receptors, sympathetic nerve endings. Their contraction leads to pupillary dilation. It can dilate up to 8 millimeters, and this is known as mydriasis. Ciliary muscles we saw in context of the accommodation. Once we talked about the change in the diameter of lens to focus at a near object. These ciliary muscles are also supplied by the parasympathetics at muscarinic nerve endings. Their contraction leads to contraction of ciliary muscles, relaxation of suspensory ligaments, and the lens becoming more globular that helps in accommodation. We discussed that in detail previously. Lastly, the ciliary body epithelium we discussed in the context of aqueous humor in our first session. These are stimulated by sympathetics, which stimulate the secretion of aqueous humor. So if there is glaucoma, what you can do is you can give antagonist to these beta receptors to decrease the secretion of aqueous humor. Either give beta blockers or give parasympathomimetic drugs to decrease the intraocular pressure in glaucoma. I hope it's understandable. Moving on towards the reflexes of eye linked with the autonomic nerve supply. Again, the details we will be much more having in details in your cranial nerve anatomy. For now, let's have an overview. This is pupil and this is its pupillary light reflex. Whenever light is shown on the pupil, the afferent nerve of the limb is stimulated. Here, the afferent nerve, which is carrying the message, is the second cranial nerve, optic nerve. Optic nerve, optic chiasm, optic tract. From optic tract, with instead of going to the lateral geniculate body, few of the fibers they separate and they go to the pretectal nucleus that we discussed previously. Here in the pretectal nucleus, they then communicate with the parasympathetic nucleus of the third cranial nerve. This nucleus is known as Edinger-Weisspol nucleus. From here, the preganglionic parasympathetic nerves arise and synapse with the autonomic ciliary ganglion. From here, the postganglionic parasympathetic nerves arise and they cause the pupillary constriction. Whenever you enter into a very illuminated area, your eyes pupils will constrict. One of the manners by which there is light adaptation. So this is the reflex behind it. The fibers from the optic nerve leaving at the level of optic tract, going to the pretectal nucleus, and from the pretectal nucleus they go to the Edinger-Weisspol. One other important thing that you cannot miss over here is from pretectal nucleus, the innervation is towards the Edinger-Weisspol nucleus of both sides. Even if the light is thrown only into the right eye, the pretectal area will stimulate the Edinger-Weisspol nucleus of both the right and left side. Ultimate resultant will be activation of ciliary ganglion and parasympathetic supply to both the eyes. So you will find pupillary constriction not only in the same eye but also in the opposite eye. This is called as light reflex, and constriction in the other eye, this is known as consensual light reflex. Consensual light reflex. Preganglionic, postganglionic, pupillary sphincter muscle and pupillary constriction. Very simple. Light falls into eye, pupils constrict of both eyes. If you remember, we had seen this diagram in our first session too, showing that this is the ciliary muscle. This is suspensory ligament, which on other end is connected to the lens. Lens themselves are elastic in nature and they want to be more and more globular. But because of the tension in the suspensory ligaments, they are kept in a flatter position as shown over here. Until the ciliary muscles are relaxed, these ligaments will be taut and lens will be flattened. If there is parasympathetic stimulation, the ciliary muscles contract, the ligaments, they become relaxed and the lens become more rounded. That increases its refractive power. This phenomenon is known as accommodation. We discussed it in detail in the first session. How does this accommodation now occur? When the fibers from the optic nerve reach the visual cortex, if you are looking at something near to your eyes, for example, you are trying to read something in your phone. When the message reaches the visual cortex, some of the fibers go back to the Edinger-Weisspol nucleus from the secondary visual areas. Visual area 18 and 19. Message will go to the Edinger-Weisspol nucleus, and that Edinger-Weisspol nucleus will stimulate the oculomotor nuclei of both sides, causing stimulation of ciliary ganglion and stimulation of the contraction of the ciliary muscles, which will ultimately increase the curvature of the lens, which will help in increased accommodation. Among children, the accommodation can lead to increase in the refractive power of lens by almost 14 diopters. So how it was started? By this stimulation of the Edinger-Weisspol nuclei by the visual cortex, which led to bilateral stimulation of the oculomotor nerve nuclei. Other than the Accommodation, once you are looking at something close to you, you're focusing on nearby object. Other than accommodation, two other events also occur. One is convergence, both your eyes move medially, medial recti, and number three, there is pupillary constriction. Again, our parasympathetic supply to the pupillary constrictor muscle. If we see all these three in combination, this is known as near response. Concluding our lecture at the pupillary abnormalities, we have seen the autonomic innervation of eye, the accommodation reflex, and the light reflex. Now we can have a look at the abnormalities in these. Before moving on to that, we have to discuss one more thing, which is the sympathetic nerve supply to the eye is coming from the cervical sympathetic trunk. Upper thoracic segment of spinal cord, cervical sympathetic trunk. This is superior cervical ganglion, which is ultimately supplying the sympathetic nerve supply to the eyes, which are meant to increase the aqueous humor production and number two, cause the pupillary dilation. This is the pretectal region, Edinger-Westphal nucleus, from which the preganglionic sympathetic nerves, parasympathetic nerves, or third cranial nerve arise. This is ciliary ganglion. From here, the postganglionic parasympathetic nerves arise. This is the cranial nerve 2, which is the afferent nerve for the accommodation and the pupillary light reflex. So let's move to the first abnormality of pupil, Argyl Roberston Pupil. This is the abnormality in which the light reflex is absent but accommodation reflex is still present. When can this happen? For example, if there is an abnormality only in the pretectal region, the reflex arc of light reflex will be interrupted. Whereas rest of the visual pathway is fine. Once the message reaches the visual cortex, the message will directly go to the Edinger-Westphal nucleus and rest of the limb is fine. Means that accommodation reflex can still occur. If this condition occurs, that accommodation reflex can occur and light reflex is lost, this is known as Argyl Roberston Pupil, and mostly the reason is damage to the pretectal area, specifically seen in the neurosyphilis. Another pupillary abnormality, relative afferent Marcus gunn pupil. Its name indicates there is something wrong with the afferent nerve. If the afferent nerve supply, let's have a look over here. Afferent nerve on one side is damaged. For example, right pupil. And light is thrown into the right eye. There will be no initiation of the light reflex. But left eye is normal, left optic nerve is normal, left afferent limb is normal. If this initiates the light reflex, it will not only constrict the pupil of the left eye but also of the right eye, because this has a bilateral activation and the efferent limb of this reflex is normal for the right eye too. So the condition in which direct light reflex is absent but consensual light reflex is present is caused because of the damage to the afferent limb only in one eye, and this is known as Marcus gunn pupil. Horner syndrome is damage to the sympathetic nerve supply to the eye. For example, can be caused by a damage at the level of cervical sympathetic trunk or superior cervical ganglion. What will happen? That the pupil will react sluggishly to the light but better to the, this is Horner syndrome. What will happen? That the pupil will be unable to dilate because the supply to the dilator muscles was coming from the sympathetic nerves. So the pupil will be continuously constricted. There will be meiosis, ptosis, drooping of the eyelid, anophthalmos, apparent anophthalmos, half of the face will be having absent of sweating. These are the other functions of sympathetic nerve supply to the face. So it will be a combination, a constellation of symptoms: ptosis, meiosis, anophthalmos, and hydrosis, caused because of the sympathetic nerve supply damage to the eye. And we stop our discussion over here. Rest of it you will be discussing with in context of your cranial nerves for physiological point of view. Three pupillary abnormalities, good to know: the Argyl Roberston pupil, relative afferent or microscopic pupil, and the Horner syndrome. That was all from my side for the special senses, the physiology of optics. If you have any questions, you are more than welcome to please comment under the video, and do not forget to watch our next videos. I wish you all the very best. Take care.